Okay, so in chapter 13, we're going to talk about the central dogma. The central dogma is pretty much going from DNA to RNA to proteins. In our cells, we hold DNA within the nucleus. The DNA is then translated into RNA, which is transcribed into proteins. So let's talk about the structure of a DNA molecule. So here's what we know about DNA, or the genetic material of life. Mendel showed in the last chapter that we're able to pass our phenotype, the visible traits, onto our offspring via the genotype, what our genes actually have, the instructional code within our genes. These expressed conditions are called traits. Before the 1950s, scientists really didn't understand what made genes or what coded for them. However, in the 1950s, we got a really good look at what we know as DNA. DNA, or deoxyribonucleic acid, is the primary material that we pass from generation to generation. Our DNA is specific to our species, Homo sapien, and we can actually map out our genome, or a complete genetic code, based on that. Right now, we have what we call COVID-19 going on. We actually can take the genetic material, the DNA that's found within this virus, and map out its entire genome. And from there, we can see what it's closely related to. And then we can learn how to treat that from there. We can also see how it changes as it interacts with other types of DNA. DNA is very simple. It's made up of 4 basic units, and those basic units are found right here in this picture. A nitrogen base, which is adenine, thymine, guanine, and cytosine. A sugar backbone, deoxyribose is the sugar found in DNA, and it's bound together through a phosphate. So it's a phosphate, a sugar, and nitrogen bases. The nitrogen bases are held together by hydrogen bonds in the center. There are 3 major experiments that led to the conclusion that DNA really was genetic material, and those were performed by Griffith, Avery, Hershey, and Chase. So let's talk about each one of those. So Frederick Griffith started working in about the 1920s, 1928 to be specific, with 2 types of related bacteria, the S strand and the R strand. Now, this page is going to talk to you about transformation a little bit, but I want to go to the other page and talk to you about his transformation experiment. So in this picture, we can see that we have 2 different strands of bacteria. This is the R strand and the S strand. The R strand is what we call non-virulent. So when we injected this strand of bacteria into the mouse lived. And in fact, what we did is we pure cultured this, injected a whole bunch of it in here, and then we waited, and then we recollected blood samples and tissue samples from the mouse, and we didn't recover the bacteria, which means the immune system of the mouse killed the bacteria. However, there are multiple strands to all types of bacteria. This one is an S-type bacteria. This one was virulent. So this one, when you pure cultured it, inject into the mouse, let the mouse do its mousy thing, it killed the mouse every time. And at the end, when you took blood and tissue samples, you were able to recover the same strand. So what they did is they heat fixed it. So when you heat kill a bacteria or virus, you're basically heating it up or cooking it. And once you do that, the bacteria dies. It doesn't take a lot of heat to do that. And what they did is they took a whole bunch of that heat-fixed bacteria or heat-killed bacteria and injected it into the mouse and realized, yeah, it didn't do anything. Well, that makes sense. There wasn't any of the genetic material. The bacteria was dead. We weren't able to recollect anything from this experiment. However, when Griffith took this heat-killed S-type, remember the S-type was the virulent type, and he bound it to the R-type, he put both of them in together at the same time in the mouse. Something crazy happened. We had what we call transformation occur. Basically, within this, we have this non-virulent, or these 2 non-virulent things. These will not kill the mouse. They were injected. And what happened is the S bacteria and R bacteria combined. And they went through something we called transformation. And the genotype actually changed back into the virulent S strand. And so they actually recollected that. And so that was pretty cool that transformation occurred. It transformed into something else. But how did it do that? That wasn't really clear until the 1940s. Avery took Griffiths transformation experiment and said, okay, what did this? RNA, DNA, or proteins? Avery used some enzymes to destroy some of the molecules in the heat-killed S bacteria and found that some of the proteins were missing. And so let's look at what he did with that is he found out that the bacteria were missing proteins and RNA. They were not able to transform the R cells into S cells. So we weren't able to go through that transformation process. So bacteria were missing the DNA and they couldn't do the transformation back into this virulent strand. Scientists concluded that the DNA is responsible for the transmutation that occurred within the bacteria. And that is true. Transformation occurs because of bacteria. It actually transforms it into something else. But then we have Hershey and Chase's experiment with bacteriophages. Okay, so bacteriophages are a specific type of virus. They're not like COVID-19. COVID-19 is a envelope virus. That's a type of virus that forms an envelope out of your nuclear membranes, and so it's hard. It disguises itself. Bacteriophages will only infect bacteria cells. That's it. But bacteriophages are really simple. They're made up of protein and DNA, and these two molecules basically pass the genetic material, and they inject it in, and it goes from there. So let's look at what happens with a bacteriophage. So these little guys right here, these are bacteriophages. Basically, they'll land, and then they have a needle that comes out here, and it will inject the bacteria, it'll inject the bacteria with DNA. And so you can see two things. One, they've put tracers in here. These are radioactive molecules that we can trace. So they've actually taken the capsid, which is the protein, and they put it into a radioactive sulfur. And then here, they took a radioactive phosphorus and injected it into the DNA. They wanted to use two separate tracers so that they could detect them afterwards. And then they just let the bacteriophages do their bacteriophage thing. They land onto the bacteria, they inject their DNA into the bacteria, and then they fall off. And what happens is their DNA will interact with that circular plasmid that we have in here, and it actually becomes part of the bacterial DNA. Now, the cool thing about that is when we started looking for this, so one, we collected all these capsids, and we found that there was no sulfur detected in the cells. They were just in the sulfur capsids that were just released. They were found within the system. Not really that great. But when we did it for the ones that injected the phosphorus, we found that there was radioactive phosphorus within the cells. So when we centrifuge this out, centrifuging basically spins it around really fast and it'll separate, and the heavy stuff go to the top and light stuff goes to the bottom. So we could separate those two out. And they found that there was phosphorus detected in there. They found out viruses pass on DNA and not proteins into the host. And the DNA is what causes the host cell to replicate the new viruses, because viruses can't replicate on their own. They actually have the host replicate more. So this, if we let this go, this whole little cell would fill up with bacteriophages until it was so full of bacteriophages, the cell bursts and bacteriophages go free. But let's talk about the shape of DNA. In the 1950s, you probably heard of these two guys, Watson and Crick. They're famous for developing the structure of DNA and understanding that it has a double helix structure, and it's kind of wound like two parallel strands, or it looks like a staircase. So they also said that parts of the nucleotide, a nucleotide basically is broken up into three groups: a phosphate, a 5-carbon sugar, and a nitrogen base. So that nitrogen base could be adenine, guanine, cytosine, or thymine in DNA. And then our sugar is deoxyribose, and then we have a phosphate group that's left unchanged. The information of DNA is contained within the base pairs, and the order in which your base pairs are found is what we know as the genetic code. So there's 4 letters that make up your entire genetic code. A, T, G, C. A will always go with T. G will always go with C. So something cool about that is these nitrogen pairs are broken up into 2 groups, purines and pyridines. So adenine is a purine, which will only bind to the pyridine thymine. And so if we look at this here, you can see that there are 2, a little bit more complexity to these here, but these base pairs, here's our deoxyribose, but our phosphate group's just a little different. And again, a cytosine will only bind to guanine. So they'll actually complete each other like a puzzle, which is really nice that they'll bind that way. And that we can correct that pretty easily. So our base pairs will always find that. So if we have a complementary pair, A, T, T, C, G, G, the complementary pair to this would be T, A, G, C, C. It's just the opposite. They'll pair up together with hydrogen bonds holding them together. Hydrogen bonds are great because they're strong, but they're easily broken and rebound. Broke, rebind, break, rebind. It's kind of like tape that never runs out. So Watson and Crick did this experiment and proved the DNA structure, and they've won a Nobel Prize for this. But it was off the backs of a few people. One of them is Chargaff. Erwin Chargaff is a biochemist that made observations about DNA. Basically, he said, hey, if you have 20% adenine, you have 20% thymine within a section of DNA. And then you would have 30% of cytosine and 30% of guanine. They'll equal each other out so that your sample that you take will equal to 100%, but the nitrogen bases will be opposite of each other, equal in comparison. That was really beneficial to know which one paired with which. He took hundreds of samples and found the same results every time. That's amazing. Here's the one that's kind of frustrating. Rosalind Franklin was an undergraduate student in the 1950s. She developed a very high-quality X-ray diffraction image of the strand of DNA. But because she was a woman and not a doctor, supervising Wilkins kind of didn't give her credit. She wasn't actually given any credit until much, 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 much later. We're talking in the 2000s before she actually received the credit that she deserves for this. But her X-ray diffraction image of DNA actually allowed for Watson and Crick to come up with the 3-dimensional model of the DNA. Without that image, we would still be looking at a boring, flat, 2-dimensional model. Probably not. We have a lot of genetic technology now since then. And so these all contributed to what we know as the structure of DNA and a better understanding of what DNA is. Until next time with replication of DNA.